Hello students, I'm Gauri Shinoy. I hope you all are well students. Be at home, be safe. Today we are going to start with the first chapter, that is cell, the unit of life. Students, you all know that all the living beings, it may be plants or animals, we all are made up of cells. So it is the basic unit of our entire body. Okay, all cells then combine together to form tissues, similar kind of tissues performing same type of function, combine together to form organs, then all the organs doing same type of function, they combine together to form the organ system. So this is how the development of a living organism takes place. So to start with, let us first see the definition of cell. So it is defined as a fundamental, structural, and functional unit of all living beings. That means it is the important and functional unit of all the living beings, including plants, as well as bacteria, certain unicellular organisms, etc. It has independent existence and perform various functions of life. So cell can exist independently without taking help of other organelles, okay, or other it can exist independently, and it performs various functions of our life. So all the activities, all the different processes that are happening in our body takes place or are it takes place inside our body with the help of the cells. You all know that cells are microscopic. They are very, very minute. We cannot see them with our naked eyes. So to have a look at the cell, to study it in detail, we need a microscope. Now, if you consider our body or an organism, the skin, brain, muscles, take an example of bone, all these are made up of some or the other type of cells. If you take an example of skin, okay, you know the outermost layer, which we call it as epidermis, it's itself, it is a tissue, but it is made up of cells. Even in our brain, we have brain cells. Okay, our muscles, they are, it is also a type of tissue, but it is composed of cells. So it is a basic unit of all the different tissues and all the different organelles. Okay, even bones have osteocytes, or what we call it as osteoblasts. So that is a type of cell that is present in bone. Okay, so all these cells which are present in our body, they have different functions, they carry out different functions inside our body. Okay, then life begins from a single cell. Okay, we human beings, we all are developed from the egg inside the womb of a mother. So that egg is nothing but it is a single cell. So even we are developed from the single cell. All the higher organisms, they are developed from, initially the development starts from a single cell, and then it goes on multiplying, division takes place, multiplication takes place, and further it develops into a fully grown organism. Then coming to, before studying cell in detail, we need to know how the invention of microscope took place. Okay? Anton van Leeuwenhoek was the scientist who constructed first microscope with a single biconvex lens, and such type of microscope was then known as a simple microscope. So what you can see here in the slide, this microscope is a type of simple microscope. As you can see, it has a single biconvex lens. So the first ever microscope that was constructed, it was constructed by Anton van Leeuwenhoek. Then Robert Hooke, he developed a microscope using two lenses. So after a few years, there was a development of a more magnified microscope, or a more developed microscope, in other words, you can say, which had two lenses for achieving greater magnification. So as the simple microscope had only one biconvex lens, the power of magnification was little less. So what Robert Hooke did, through his studies, he developed a microscope which had two lenses, so that the magnification of whatever object we are observing under the microscope would be more. Coming to discovery of cells. In 1665, scientist Robert Hooke used a microscope to examine thin slices of cork. So Robert Hooke used this microscope for observing a thin slice of cork. Cork is nothing but a plant material. He observed that cork is made up of tiny hollow compartments. So when he observed this cork tissue, this is a cork tissue under the microscope, he saw that it was made up of many tiny hexagonal compartments. They appear to be like honeycomb structure, or you must be knowing that in a monastery, all the monks have those small rooms. So they appeared like the small rooms. So he named those rooms as cells. Plants Hooke observed were actually dead. So this cork, or the plant material which was observed by Hooke, was a dead material. He was actually observing cell walls and empty spaces. As this material was dead, what he could observe is only the cell walls, the periphery of the cell, and the empty spaces in between. Okay, so this was a microscope which was developed by Robert Hooke. As you can see, it used to work on, it has a stage. Here we can keep our sample. This is the lens on which the light falls, and this microscope used to use the light from which was emitted by an oil lamp. So this is an oil lamp over here which emitted light, which will fall on the lens, and then we can see the object which is placed on the stage properly. After a few years, there was a discovery of electron microscope, which gave us very high magnification. It gave us better magnification of all the objects that we could observe under it. So let us see a comparative study between light microscope and electron microscope. As you can see in the picture, this is a light microscope and this one is an electron microscope. Now, let us not look at the cost. Now here in light microscope, they use the visible light, okay, from any of the light source that is used for observing the object which is kept under it. Whereas in electron microscope, it uses electrons to scan specimens and to create the magnified images. So here in this microscope, they use electrons to scan the specimens, whereas in light microscope, we use visible light. The magnification of light microscope is usually between 1000 to 2000 times at most. So whatever specimen you are keeping it under the microscope, magnification will be 1000 to 2000 times of that specimen. Whereas in case of electron microscope, as I told you, the magnification is very high. It is up to 50,000x. That means the sample is magnified 50,000 times. Here in light microscope, it has a low resolution. That is why whatever object or whatever specimen you are observing under the light microscope, the resolution, the detailing will not be there. It will be there, but it will not be much detail. You cannot study the structure in much detail. Okay. But if you want to study any specimen in detail, you need to use the electron microscope because it has got higher resolution than the light microscope. Light microscope always produces 2D images. So the image that is observed is in the form of 2D structure or a 2D image. Whereas in case of electron microscope, the image that is produced is clear and it is a detailed 3D image. So advantage of light microscope is better for observing living specimens. So here we use light microscopes for observing the dividing cells, for cell movements, whereas electron microscope Specimens has to be held in place, unable to view living specimens. So with the help of electron microscope, we cannot actually view the living specimens, and they have to be held on place. We just can't keep on moving the specimen which is kept under the electron microscope. Okay, so this was a comparative study, students, of light microscope and electron microscope. So from this, we have studied that electron microscope gives us much magnified view of whatever sample we are observing under it, whereas light microscope give us a comparatively lesser resolution or a lesser magnification of the object. Basically, in our school level, we use the compound microscope or the light microscope because we don't have to study the specimens in detail like the scientists do. Okay, we just have to observe the organelles inside the cell or the cytoplasm or the cell wall, that's it. Next, coming to cell theory. Okay, so Robert Brown, he was a scientist who discovered the nucleus in 1833. So he was a scientist who first discovered the nucleus inside the cell in the year 1833. Matthias Gilden was a German botanist. He, in 1838, had discovered that all plants are composed of cells. So through his studies, he has proposed that all the plants which are present on the earth, they all are made up of cells. They are composed of cells. Theodor Schwann was another scientist. He was a zoologist basically. In 1839, he also discovered that animals were made up of cells. Okay, so he also was undergoing through different studies on animals, and through those studies, he came to the conclusion that unlike plants, even animals are made up of cells. Different type of animals are made up of different type of cells. Okay, so later, Rudolf Virchow in 1856, he was a German physician. He stated or he postulated that cells only come from another cells. That means new cells always arise from the pre-existing cells. Okay, so new cells are continuously developed inside in our body, or they are formed inside our body through the older ones, through the old cells that are already existing. So the cell theory, which was proposed by the scientists Schwann and Sheldon, it states or it explains three important points. They are: the cell is the smallest unit of structure of all the living beings. So for all the living beings, may it be plants or animals, cell is the smallest part or the unit of structure that is present inside the body. The cell is a unit of function. That means all the functions that happens in our body are because of the cellular activities that are taking place inside our body, and all cells arise from pre-existing cells. As I told you, a new cell is always formed from the pre-existing ones. See, students, in our body there is continuous um, warning out of the older cells. When the cell becomes old, okay, they die, they are warned off, and then the new cells take place of the older ones. So always the cells come from the other cells. Okay? Now let us see why cell is known as a structural unit of life. Okay, because if you take any organism, okay, or if you take any part of any organism and observe it under the body, a thin section under a microscope. Okay, for example, we are taking a thin section of a leaf and observing under a microscope. What will you observe? You will observe that it has got the cellular structure. You can see the hexagonal cells that are present inside it. That is why even if you take a thin section of a frog or maybe any part of a cockroach and try to observe it under the microscope, you will see that even our skin, okay, if you peel out a thin section of your skin finger and observe it under the microscope, you will see the hexagonal cells that are present inside. That is why cell is known as the structural unit. Then coming to functional unit. Why it is known as the functional unit? Because the activities, the different type of processes that takes place in our body are because of the cells. For example, the movement of the frog. Okay, frog hops or it jumps from one place to another. This is due to the contraction of the muscles which are present in his limbs. Okay, and how do these muscles contract? With the help of the cells that are present in the muscular tissue. Okay, another example which I can give you about the function is the food that we eat. Okay, it is digested with the help of the enzymes. Now, where from these enzymes comes into picture, or from where they are produced? They are produced by the cells of the gut. Okay, so our gut, the gut is made up of cells, and those cells secrete certain enzymes which helps in digestion of the food that we eat. That is why cell is called as the functional unit of life. Okay, and as I told you, all the cells die and they are replaced. Okay, new cells are formed by the old cells. Then coming to, these are the different examples of cells that you can see on the slide. Okay, this one is the cell of an amoeba. This is a plant cell. It's a different type of bacteria. Okay, you can see a cell. This is a nerve cell. Then the red blood cells or the RBCs. Now, we have just studied the cells. As I told you, cells are very, very small. They are too minute. We cannot see by the cells with our naked eyes. Okay, so cells are small basically for two reasons. Because the cells are small, they have more surface area uh, than a large cell for a given volume of cytoplasm. So a smaller cell has always got a more surface area than the volume. The ratio of surface area to volume is more for a smaller cell. Now, because of this ratio, what happens is different type of cells which are present in our body, they can communicate with each other rapidly. Because the cells are small, they can communicate with all other cells or different type of cells in our body rapidly for the cell function or for a different type of functions. Second, cell has a large surface area by volume ratio for greater diffusion of substances. See, inside our body, whatever, even take an example of digestion. So whatever we eat, the food that we eat, it has to be digested, and then it has to be the important materials or the the important part that we need for our growth, the vitamins, minerals, carbohydrates, proteins, whatever is there, it has to be diffused inside our blood so that it can be sent to different parts of our body for our growth. So this diffusion is brought about only because the cells are small. If the cells are big in size, what will happen? The surface area and volume ratio will be more. So they will take more time to communicate between each other. Okay, and the diffusion also will take much more time. Then coming to cell shape, as you all know that there are different cells are of different shapes. Okay, as you can see here, and this shape is basically, it depends upon the function. The shape of the cell basically depends upon the function. Now first, as you can see here, RBC, this is a red blood cell. In human red blood cells, they are circular, as you can see, and they are biconcave. Okay, human red blood cells are circular and biconcave, so that they can pass through the narrow capillaries and transport oxygen. Okay, so why they are circular, why they are biconcave? So that, see, capillaries are very, very fine networks. Okay, they are the blood vessel, a type of a blood vessel, and they are very, very fine. So just imagine if the red blood cells are too big, they won't be able to pass through the capillaries. So to carry out its function properly, red blood cell has this particular type of shape. Okay, then At the same time, if you see white blood cells, they are amoeboid. Amoeboid means like in amoeba. They have pseudopod, like how amoeba has pseudopodia. What do you mean by pseudopodia? It is a false food, you can say, which helps in locomotion. So why white blood cells are amoeboid? So that they can squeeze out through the capillary walls, because they have to come out of the capillary wall. They have to squeeze out from the capillary wall. That is why they are amoeboid in shape. If you observe nerve cell is the longest cell that is present in our body. Why it is longest? Because it has to conduct impulses, or it has to transport messages from one part of our body to other. Nerve cells always conducts message from our brain to different parts of your body. That means from tip to toe, it has to pass messages. That is why it is elongated. It is very, very long. If you see muscle cells, they are also long and they are contractile. Why they are contractile? Because muscles, they have to pull or they have to squeeze our body parts. Okay, the movement of our arm or the movement of our legs is dependent upon this muscular cells, because of the contraction and relaxation of this muscular cells. That is why muscle cells are long and they are contractile. If you must have observed the stomata are the small openings on the lower side of the leaf. If you observe the lower side of the leaf under the microscope, you will see the stomata, which is a type of a cell. So this stomata basically have they have guard cells, okay, or stomatal pores, and they are bean shaped. They are like the shape of the kidney beans. Why? So that they can open and close the pore. Okay, they can open and close the pores on the leaves. That is why they have the shape of a bean. So from this we have learned that the shape of the cell is dependent on the function that it performs. Next, coming to size of the cell, here you can see different types of cells, and you can actually see the scale over here. Okay, it is 0.5 nanometer, 1 nanometer, 10 nanometer, 100 nanometer, 1 micrometer, and 10 micrometer, 100 micrometer, 1 millimeter. Okay, so if you see this polio virus, it is 30 nanometer in diameter. Okay, so this is the actual shape of the polio virus, HIV, or the AIDS virus. So these are the different examples of the sizes of the shell. Okay, a hen's egg, it is 65 millimeters. Okay, so it is the biggest. If you can see in this particular slide, if you see, this is the biggest cell that is present. Okay, so the smallest cell which is present is of the bacteria, which is about 0.3 to 5 micrometers. We have red blood cells, which are about 7 to 8 micrometers in diameter. The longest cell, as I told you, is the neuron or the nerve cell. Okay, and the largest cell is the ostrich egg. Okay, that is the largest cell that is present. Now we are going to study about the cell structure and functions of different organelles that are present inside the cell. Here you can see the picture of animal cell and plant cell. This is the detailed structure of animal cell, and this is the detailed structure of plant cell. Okay, animal cell, as it does not have a cell wall, you can see that the structure is irregular. Okay, whereas in plant cell, as it has a cell wall, it has got a definite structure, or it has got a rigid structure. Before coming to the different type of organelles, let us first study the type of cells. There are basically two types of cells, unicellular and multicellular. From the name unicellular itself, we get to know that the organisms which are made up of only one cell are known as unicellular organisms. Okay, so their entire body is made up of only a single cell. For example, we have amoeba, we have paramecium. Then coming to multicellular, multi means many and cellular means cells. So the organisms which have more than one cell okay, they are known as, or which have many cells in their body, they are known as multicellular organisms. For example, we have human beings, we have animals, we have plants, okay, so they all are multicellular. Now, besides unicellular and multicellular, there are two major categories of the cell. Okay, these categories are prokaryotic cell and eukaryotic cells. Okay, prokaryotic cells examples are bacteria and archaebacteria. Eukaryotic cells examples are plants, animals, and fungi. Now, let us study in detail about the prokaryotes and the eukaryotes. These are the pictures which you can see of a prokaryotic and a eukaryotic cell. First, let us study about the prokaryotic cell. It do not have the structures surrounded by membranes. Okay, so first, prokaryotic. Karyon means a nucleus and pro means primitive. Okay, so the organisms which have a primitive nucleus are known as prokaryotic organisms, okay, or prokaryons. They do not have the membrane bound organelles inside the cell. Inside the cell, if you see, there are no organelles present. Okay, there are few internal structures, like you can see cytoplasm, you can see few ribosomes, but not all organelles are present inside the prokaryotic cell. Basically, all the unicellular organisms are known as prokaryotic. Okay, their nucleus is not well developed. Then coming to eukaryotic organisms or eukaryons, they are the most complex forms of life. Okay, we all are eukaryotes. Why? Because our nucleus is well developed. If you see the structure of animal cell, we have a well developed nucleus. We have different organelles present inside the cytoplasm, which are surrounded by the membranes. So basically, all the multicellular organisms which are made up of many cells are eukaryotic. Okay, examples are animals and plants. Okay, so this is a plant cell and this is an animal cell. So as you can see, there are different organelles present both in the plant cell as well as animal cell, and they are surrounded by the membranes. Okay, for example, in plant cell, we have a cell wall, we have a cell membrane. In case of animal cells, we don't have a cell wall, but we do have a cell membrane that is present. Then you can see a nucleus, there is a vacuole, then there is mitochondria, there are liposomes, there are Golgi apparatus, chloroplast. These are different organelles that are present in the plant cell. Whereas in animal cell, you can see centrioles, you can see nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus, then you can see mitochondria. These are the different organelles present in an animal cell. So that is why we are known as eukaryotic or eukaryons. Why? Because our nucleus is well developed. Now this is the figure of a typical animal cell. Okay, you can see different organelles inside it. This is a cell membrane. Okay, this is an endoplasmic reticulum. This one is a nucleus. This is an Golgi apparatus, a mitochondria. Then we have lysosomes. These blue ones, the small ones, are the ribosomes. This is a picture of a typical plant cell, a magnified view. You can see all the structures clearly. This one is a cell wall. Then cell wall is the outermost membrane. Then we have inside the cell wall, we have a cell membrane. Then this one is a cytoplasm, and all the organelles are embedded inside the cytoplasm in case of both plants and animal cell. This is in Golgi apparatus. Then you can see Golgi vesicles. These are the ribosomes. This is an endoplasmic reticulum. This is a nucleus. So endoplasmic reticulum is basically near the nucleus. This is a nucleolus which is present inside the nucleus. All these organelles we will be studying in detail. Then this is a vacuole, a large central vacuole that is present. Okay. Chloroplast you can see here in plant cell, which is absent in animal cell. Then coming to organelles. What do you mean by organelles? From when I'm using this word, organelles, mitochondria, ribosomes, Golgi apparatus, and we have endoplasmic reticulum, centrosomes, centrioles, all these are known as organelles. Why? They have a definite shape, a definite structure, and they have a definite function to perform inside our body. Now first we are going to study about the structures which surrounds the cell. Okay, if you take an example of cell, you will see the cell membrane. Okay, this is the outer membrane of cell that controls the movement in and out of the cell. Okay, so cell membrane is the outer membrane that is present both in case of plant cell as well as animal cell, and it controls all the in and out movement. It is double layered, as you can see in this picture. This is a magnified view of cell membrane. You can see two layers of the membranes. It is selectively permeable. Cell membrane is always selectively permeable. It will allow only selected substances to pass through it. Okay, this in case of animal cell, you can see this cell membrane, and in case of plant cell, this one is the cell membrane. Okay, so basically it is made up of lipids and different types of protein. It forms a bilayer. So it is the outer boundary of the cell. It is a double lipid membrane. It is made up of double layers of lipid, and as I told you, it is selectively permeable. Now let us see functions of the cell membrane. First and the foremost function, the most important function of the cell membrane is protection. As it is the in case of animal cell, it is the outermost membrane. In case of plant cell, we have cell wall as the outermost membrane. So it helps or it protects all the inner organelles that are present inside the cell. It gives supports and shape. Okay, so cell membrane always give supports to the plant and the animal cell, and it control what enters or leaves the cell. So whatever has to enter the cell, or the substances which has to go through which has to be thrown out of the cell, depends upon the cell membrane. So it acts as a gatekeeper. Helps maintain homeostasis, ability to keep a stable environment. Okay, so homeostasis means it always maintains a stable or a steady environment. So that is also done by the cell membrane. Then coming to cell wall. Now cell wall is present only in the plant cells. It is not present in the animal cells. So this outermost layer which you can see is the cell wall which surrounds the cell membrane. This one is a cell membrane. It is made up of cellulose. So cell wall is made up of cellulose in case of plant cells. It is always strong and rigid. That is why it gives a rigid shape. It gives a definite shape to the plant cell. It is a dead layer and it is freely permeable. So what it will do, this cell wall is freely permeable. It will allow all the substances to pass through it. But inside cell wall, the next layer is the cell membrane in case of plant cell. So cell membrane is selectively permeable. So it will allow only few substances, even though all the substances have got entry into the cell into the plant cell, the cell membrane, what it will do, it will allow only few substances to go inside the cell actually. Functions of the cell wall, again, it protects or it gives support to the cell, and it gives a rigid shape or a definite shape to the plant cell. So these two are the functions of the cell wall: protection and to give a shape and support to the plant cell. Then coming to cytoplasm. Now inside this, in both the cases, in both the cells, animal as well as plant cell, inside the cell you can see that it is filled with cytoplasm. Now cytoplasm is a jelly-like substance. It is a watery fluid that is present in a semi-liquid fluid, you can say, which is present inside the cell. It occupies most of the part of the cell, and under the microscope, if you observe any cell under the microscope, this cytoplasm will be colorless. It will be somewhat watery, and it will be partly transparent. Okay, so 80% of the cytoplasm consists of water. Coming to functions of cytoplasm, now organelles float inside the cytoplasm. So all these organelles you can see them floating inside this cytoplasm or the semi-liquid fluid. Materials for growth are found in cytoplasm. So whatever is required for our growth and development, the materials that are required are always present in this cytoplasm. Besides this, the chemical reactions which are taking place inside the cell, they always take place inside the cytoplasm. Cytoplasm also expels waste through the cell membrane. So whatever is to be thrown out from the cell, it is expelled with the help of cell membrane by the cytoplasm. Okay. In the next class, we'll be studying inside the cell. What all organelles are present inside the cell? What are their functions? How do they look like? Okay. Students, thank you so much. Thank you.